Okay, good morning everyone.
Good morning, sir.
Since the camera is working, we can start. Okay, let's do it on time. Today it's third set, okay? So we have more or less on the middle of the way. So we can continue with the safety. Remember on the practical I was repeating safety, 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 safety. Okay, we have safety when we work in the laboratory. We need to know that microbiological laboratories need to respect the rules of the laboratory. But as well, we need to be sure that microorganisms that we're going to apply in the starter culture, we're going to apply as a beneficial culture, etc. This needs to be safe, because if it's not safe, how we can give something that is not safe to the consumers? Different points need to be discussed. Biogenic amine production could be a problem, virulent factor, antibiotic resistance, so all these kind of things needs to be taken in consideration. And as I wrote, safety, not all are angels. Well, lactic acid bacteria in general is considered as a safe. And very often in the articles, people put lactic acid bacteria, they have GRAS status. Well, no. GRAS status generally recognized as safe. It just says generally. It's not meaning safe. Something that one bacterial strain is safe, not bacterial strains from the same species will be safe. So safety needs to be looked always on the strain level, not on the species level, neither in genus level, neither in lactic acid like a group of bacteria, lactic acid bacteria level, because very clear examples. Streptococci, I mentioned in one, I think in the first lecture, only 3 species from Streptococcus. Streptococcus thermophilus, Streptococcus salivarius, and Streptococcus macedonicus is considered as safe. And in fact, we're having documents of International Diary Federation. They publish every like 2-3 years, they're updating this list. They put the name of the microorganisms that are considered safe and can be used in the dairy industry. So don't need to remind you, Streptococcus pneumonia, it's not as safe. I mean, the name is already showing that it's a very dangerous microorganism. Group of Enterococci associated with vancomycin resistance could be a very serious problem in the medical care. In general, Enterococci are considered as a problem. Well, in this point I want to open one little bit different topic. Very often, microorganism was isolated from some ecological environment and have his name. This is historically. Since Enterococcus fecalis was isolated from fecal samples, automatically in the first isolate been classified as a problematic pathogens. In general perception for most of the Enterococci that they are a problem. Well, not all of them. We have several Enterococcus, especially Enterococcus faecium, Enterococcus hierae, Enterococcus munti. They can be associated with the different fermented food products. And if you look the different fermented food products, especially in the Mediterranean part of the Europe and Africa, several fermented food products are fermented by the Enterococci. So not all Enterococci are dangerous, not all Enterococci are safe. So safety needs to be checked on strain level, on species level first, and then on strain level, because we can see that even some species that are considered as a safe, in some conditions they could be dangerous. End of the day, every bacterial species in the high number, it's not good. I mean, everything is good in moderation, and when they're in the right number, in the right genetic heritage, they can be considered as appropriate to be applied. So in general, you see, like when we was talking about lactic acid bacteria, you have several of them. You have Lactobacillus, Lactobacillus plantarum, Ocarnobacterium maltaromaticum. We have Lactococcus lactis. They are used in several fermented food products. Enterococcus fecalis, Streptococcus infantarius, Streptococcus thermophilus. You see already you may be having idea separating them safe and not safe. Leuconostoc mesenteroides, Pediococcus. So most of this on the left side actually consider it as a safe, but they consider it as a safe, but don't mean they're safe. So they need to be tested. Interesting story, it's about Streptococcus infantarius. It's a strain that is associated with, was isolated originally from dairy products from, let me just think, Western Africa, and later being actually isolated from dairy products in Brazil, most probably by the slave trade in the past, trading some traditional products in the past. There was kind of transfer this strain on the other side of the Atlantic. But interestingly, this strain was become predominant in one dairy plant. Every time when we was trying to isolate something, we always have this strain. And interestingly, he's quite highly pathogenic, have several virulent genes, but in the same time he's producing bacteriocins. So now it's coming the question, we're having the pathogen, we're producing bacteriocins. So bacteriocin production is beneficial or virulence factor? Depends on the property of the strain. If you have the beneficial strain that can produce the bacteriocin, this is a beneficial. But if it's a pathogenic strain that can produce the bacteriocin, in this case, bacteriocin could be considered as a virulence factor because it's helping to this strain to colonize the environmental and to take over the other strains. So you see in the well, but I mean, look in your traditional story, everything is like good or bad depends on the situation. So it's nothing, it's exactly white or black. Everything is something in between. Same thing with the bacteriocins. When they're carried by the pathogen strains, they can be considered as a virulence factor because they will give the opportunity to pathogen to be even more pathogenic because he will take all over the environment and will continue to exist and to make his bad parts. So, story about the Streptococcus, as I mentioned, some of them, Salivarius, Macedonicus, and Thermophilus, are considered safe. They're part of the variety of dairy products. Most probably, in the evolutionary perspective, they lost several of their virulence genes because since they're in the dairy environment, they don't need these virulence genes to survive. And in the nature, in the evolutionary process, if you don't need something, you will lose it. Because, I mean, Streptococcus thermophilus needs to be hemolytic. He never will be in the contact in the humans. Well, it's coming in the human cells because of the yogurt, but again, in the natural perspective, he's in the dairy environment. There is no need to be hemolytic. You don't need this activity. But the rest of the Streptococcus, they could be alpha hemolytic, they can be beta hemolytic, and I mean, don't need to show you all these examples. The Streptococcus mutans, Streptococcus mitis, they are associated with some dental problems, principle associated with caries, Streptococcus pneumonia, then you have even Streptococcus pyogenes, that is even worse. And all of them, they are associated with several problems. But just a good example of this that we cannot judge just by the name. We need to know exactly specificity of every species, every strain, and then we need to try to see what is, if they are good or bad. Virulence factors, they can be associated with the beneficial and, okay, I think I lost that. It's working now, no? Can you, sorry, I moved something and the system, you plug and, deplug and plug and this work always. Okay, so the virulence factor, they can be everything that actually can help the microorganism to perform his bad behavior. We can consider it as a virulence factor. This could be associated with addition because when the bacteria adhere to some places, they can actually facilitate their negative influence. This can be invasion, this can be competition for iron and nutrients. Microorganisms need microelements and they can do everything to have these microelements. In fact, hemolytic activity is one of the ways today to destroy the hemoglobin and to have enough iron for their metabolism. Resistant to of host immunity. They will try everything to block the immune system because the immune system will try to reduce them, or even the secretion of some toxins. So, in fact, virulence factors, they can be associated with the microorganisms, they can be associated with the different bacteremia, endocarditis, and it's interesting that even in lactobacillus, that is considered as generous and safe, especially with the pathogens, with the patients with some clinical problem. They can be even worse. But even the safe microorganism, they can be in some cases associated with some carrier genes. Presence of these virulent genes, antibiotic resistance, easy to check because, I mean, I was already discussing with you this. This day, everyone is want to do the biomolecular work. So PCR. So by PCR, you can easily detect the virulent genes. But are you detecting everything? All the virulent genes are described as a PCR product. Well, this is why even antibiotic resistance, we don't know. Okay, we have a vancomycin resistance. We have like 6 or 7 genes associated with vancomycin resistance, but maybe it's another genes that are not described. So always, you can do the PCR, you can detect the gene, because in this case, you know, yes, strain having virulence factors, have the antibiotic resistance. They don't express, but they carry this in some condition, they can start expressing. But always it's good to do the biochemical and physiological test. They're a little bit more work, but again, they will give you the real picture. Because how you can be sure that it's not existing another virulence genes, another antibiotic resistant genes associated to vancomycin resistance that no one was described. And you're still detecting when you plate resistant to vancomycin. So you take checking the presence of these different genes. They're not present, so why we have this resistance? So go deeper and discover the new gene that is associated with this kind of antibiotic resistance. Biogenic amines, they can be another serious problem because biogenic amines, there's a natural process. They are nothing strange for the microorganism. It's a natural metabolic process. You have the amino acids, they will decarboxylate and they will produce these biogenic amines. Everything's okay from the point of view of the microorganism, but from our point of view, this is not good because they can have toxic, they can be responsible for toxicity. They increasing of the amount of biogenic amines can even interfere with your behavior, can interfere, can give a headache, can give even poisonings. And it's becoming a problem. Okay, if they produce the biogenic amines, what do we need to think about? It's worth this story when we have the live microorganisms, because if you produce something and you have the production of biogenic amines, but then you cook the product, there is no problem, because most probably even the cooking can maybe destroy some of the biogenic amines, maybe. But again, since you kill the microorganisms, you're not going to produce more. Problem with probiotics and the fermented food products is you have the live organisms, and they can continue producing the biogenic amines during the shelf life. So you pack cheese, and you have some enterococci then inside this cheese, and they can continue working, and they will continue producing biogenic amines. It's interestingly that we have some regulations around the world. Okay, sorry, first, before the regulation. So, normally the production of biogenic amines could be because of the carboxylation. So you have the amino acids, you're losing process of the carboxylation, you have formation of biogenic amines. This is more common way of their production, but you have reductive amination. So in this case, actually, you having including amino group, and you have formation of the biogenic amine. You can have transamination. You can have hydrolysis. So you see, it's several ways and pathways to actually microorganism. And since the biogenic amines is kind of part of this network of metabolic network in the microorganism, you cannot escape. Well, some of the microorganisms, let's say Lactobacillus plantarum, they're normally considered as a negative. They very rarely can be detected by biogenic amine production. If you look to enterococci, they always is associated with production of, now you pick me, which one was? There was one of the amino acid that is always decarboxylating. I will check and I will let you know in the next lecture. But it's something typical in enterococci. You can always have some of the biogenic amine production. Then Lactobacillus curvatus is always as well, it's typically Lactobacillus curvatus to have, because it's part of their metabolism. If you take Escherichia coli or Salmonella, or you can always detect production of biogenic amines. And normally when you do the test, you know, always you need to have the positive and negative control, right? To be sure that your experiment is working. So if you do the biogenic amine, always put one Salmonella as a positive control, always will give. And the test is quite easy because first of all, you need to stimulate the gene, because maybe gene is there but it's not expressing. So how it's work? You culture your microorganism for like 3 or 5 or 7 constitutive renewing every day in the normal growth media. Could be MRS for lactic acid bacteria. And then you add extra amino acids, because you want to stimulate this organism if they have the genes to start expressing this gene. And then you can add a pH indicator in the media, because biogenic amines, what will be the pH? Low or high? Amines, high. So you acidify your media, you culture your organisms, and if they start producing biogenic amines, since they will change the pH from a neutral and actually even like going up to 8, 9, 10, they will change the pH, and this will change the pH indicator. And even you can see the change of the color of the media from yellow to purple. And then you can make a plate as well when you put amino acid that you're targeting to be transformed to appropriate biogenic amine. And then you can actually see when you grow the culture on the surface of the plate, you can see the change of the color of the media around the colonies. It's getting from yellow to purple. It's very easy test, well, taking time, take about 1 week every day, new one, but at the end of the day it's nothing too difficult. So we have different amino acid precursors for the biogenic amine. You can have histidine, tyrosine, tryptophan, lysine, phenylalanine, arginine, ornithine. And via these processes that can appear normally, they're carboxylation, you can generate biogenic amines like tyramine, histamine, tryptamine, cadaverine, phenylethylamine, agmatine, and putrescine. So appropriate amino acid, they can go to all these biogenic amines. Well, not all of them actually are too bad. Well, in general they consider it as a problem. Some of them a little bit more, some less. But the question is, we need to know what's the level. Normally, this of the biogenic amine histamine, that is from derivative from histidine. Histamine is normally considered like one of the most problematic. It's always been targeted in the food industry. And then having a different rules. I mean, if you look the literature, if you look the different countries, they have some variation, how much could be. They have different variation for fish and fish products. The governmental international organization, independent research, they was having some kind of trying to standardize. They don't really have the consensus, but it's important thing that they was like, you see, from 50 to 500, more or less, they put in most of them. Well, I think if you have this biogenic amine, rather check your production line and don't allow microorganism that can produce this biogenic amine, histamine, don't allow it to be used as a starter culture, because obviously will give the problem. You don't want this. And actually smell bad. You know how fish stay on the sun for 1 week? Because actually some of the time it's actually production of histamine. So needs to be avoided. Another problem on the safety of the on the lactic acid bacteria is the fact that lactic acid bacteria can produce lactic acid. Well, lactic acid is considered as a good thing, but then it could be D-lactic acid. It's not only L-lactic acid. And now it's coming an interesting point. Accumulation of the D form is actually it's a big problem for babies and small children, because they cannot actually effectively metabolize this acid, and it will accumulate and will make acidosis. And you don't want your baby to be with colica and high acidosis because obviously it's not good for your child. For adults, normally our organism is able to compensate to some extent. It's not such a big problem, but for the babies and small children it's a big problem. And this problem actually started quite recently, in 1979, or et al. They described that the lactic acidosis, it actually can be associated even in some bowel disease and neurological manifestation and severe metabolic acidosis. And it's more and more winning attention because for several lactic acid bacteria, production of lactic acid together with L-lactic acid is a completely normal physiological process. But again, microorganisms are not here to serve us, they're for their own purpose. So when we select microorganisms for our work, we need to try to check to don't select this one that predominantly produce the acid, and or even the equal quantity. We need to one that was much more with L-lactic acid. We was doing a test in our laboratory. One of student in Brazil, we have it's called scientific initiation. Normally, it's students from undergraduate course. They lab and they do the project, about 1 year project, and they can do some laboratory work. They can do some research under the control of some seniors. And in this case, Isabella, she was needed to check different probiotic products. So we collect the probiotic products, predominantly interested in this that having Lactobacillus reuteri, because we needed Lactobacillus, or actually Limosi Lactobacillus reuteri. We needed a reuteri for some different purpose because we wanted to produce reuteri in the lab, and we needed a strain that could be produced. So we took 10 different probiotic products from the market. We was having 2 from Europe, 2 from Portugal, from US, and 6 from Brazil. Interestingly, 2 of the products there was nothing, no live cells. And to be a probiotic needs to be a live cells in the specific number. So 2 of them, nothing. Well, I was quite surprised. No, actually it's not surprising. We know that several of the products on the market sometimes cannot match with the exigence for their specificity. So this means the controlling organs needs to be more strict with these companies. But interestingly, we detect reuteri. Most of them are really reuteri, no contamination. 2 products were with contamination. Okay, but this is a different story. Now, we checked the production of L and D lactic acid. And you can see some of the isolates. We have several of them. Blue, it's the lactic acid. Yellow, it's L-lactic acid. Some of them was okay. I mean, this F1, very high amount of lactic acid, almost nothing of D. It's completely normal to have some quantity of D. But something that actually is worrying me, most of them was the lactic acid was more than a lactic acid. And worst thing, some of these products being recommended for the babies. So no one was checking, or was negligence, or what's going. So this is a good example that it's your responsibility as a scientist and developing new products and putting something on the market to be sure that you're not going to provide nothing wrong for the consumers. Virulence factors, continue with them. They are molecularly expressed and secreted by pathogens. Normally we say pathogens, but they can be secreted by lactic acid bacteria. And they could be helping these organisms to colonize the specific ecological environment. They can immunoevasion to help them to be more effectively to reduce the immune response. Immunosuppression, inhibition of the host immune system response. They can be entering inside the cell, so acting on the cell surface and mess with the metabolism, obtain nutrition from the host even. I mean, imagine you have the microorganism that can be much more easily take iron, and iron will finish in the environment. So this microorganism will take over the other one. Virulence factors are very often responsible for causing disease, but it's not necessary. They can be just competing with the other microorganism. And normally it's associated with capsules and endotoxins, and very often they can be associated with the plasmid. Some of these factors can be located on the plasmid, so can be easily transmitted to the other. When we're talking about transmission of the virulence factors, let's say antibiotic-resistant virulence factors, everyone was complaining if the virulence factor is from the plasmid, that can be easily transmitted. Who was trying to do the cloning of the gene on transmission of one plasmid from another to another bacteria? Someone was trying in the lab. It's not that easy, in fact. You need to be several conditions, you need to put the antibiotics, you need to do several work, and it's not so easy. But unfortunately in nature it's happened. It's happened much more easily in nature than in the lab. There was a very interesting story about performed with lactic acid bacillus rhamnosus as a probiotic. You heard about Rhamnosus GG strain? Okay, it's one of the very famous probiotic strains all around the world. Same company producing and selling all around the world. It's quite considered like a good probiotic strain. They say it's a panacea that solves all the problems. I don't believe, because there is no probiotic that can solve all the problems. But okay, they have several studies and showing that it's a good strain. Interestingly, they was making a simple test. They was feeding the mice with the salmonella and with the rhamnosus. And in a couple of months, they discovered the genes from salmonella inside the rhamnosus. Rhamnosus. Because always confusing with rhamnosus and reuteri. So they was using rhamnosus and salmonella, and in a few months they discovered naturally in the gut of the mouse, when the two strains been together, they had a transfer of the genes from salmonella to rhamnosus. And the point is that we don't want salmonella, right? It's a pathogen, everyone agrees with this. But again, rhamnosus is considered as a probiotic. It's a strain that is selected because of his adherence, his behavior. This strain loves to be in the human gut. So he will be difficult to cure out. It will stay there. But the something that worries, you see in the natural process, the genes, virulent genes from salmonella was easily transferred to rhamnosus. In fact, you can create rhamnosus with pathogenicity. So it's a very interesting point. This is why normally when you work with the strains in the probiotic area, you want strains that don't have plasmid. Because if the strain carry plasmid, these plasmids can be easily transmitted to the other strain, much more easy than genes from the bacterial chromosome. This is completely clear. Plasmid, they can move from one to another. This horizontal gene transfer is nothing unusual. But again, you don't want to transmit one gene from one to another strain. This is why when we have antibiotic resistance, we don't want the strain probiotic to carry antibiotic resistance, because the genes of antibiotic resistance from probiotic can be transmitted to the pathogens, and then you make pathogens even more worse and more stable and more pathogenic. Different factors can be associated with, let's say, hyaluronidase production, different proteases, coagulases, lipase, desquibunical NAS, and enterotoxins. Some examples of virulence factor in Streptococcus pyogenes. You see, Streptococcus thermophilus is a good guy, but Streptococcus pyogenes is an extremely bad pathogen. Could be M proteins, lipoic acid, hyaluronic acid, or even capsules, invasions such as streptokinases, streptodinases, hyaluronidase, etc., etc. So in the nature, don't miss virulence factors. And the pathogenic organism, they collect them and they have them and they will keep them. And they will try to spread them. So this is why we need to be sure when we work to don't have nothing that then will transmit. Antibiotic resistance is another problem, because working with the probiotic microorganisms, we don't want to spread antibiotic resistance. And if we have the genes of antibiotic resistance, we need to be careful with these strains. So in fact, naturally selection of antibiotic resistance pathogens as enterococci or even streptococci can occur. And it's very important, again, remember, when the penicillium being discovered, everyone was happy. Okay, we finished with diseases. Not at all. Because how would it be appropriate to say, not on control, on behavior, human behavior. Start using antibiotics for everything with the idea to protect. In fact, the best intention, the shortest way to the hell, right? So they wanted to protect, they wanted to make it everything better, but they in fact created Frankenstein. Because they start using antibiotics as a growth promoter in the animals farming. Well, it's good to protect your animals from the disease, but not necessary, and you actually start selecting, because you're giving them the antibiotics, and you start select these antibiotic resistant strains to be more and more powerful. Then was using antibiotics in the fish farming. Well, you put in the water, but this water is going where? I'm not going to drink this water sometime later. This water is not going via the normal draining system and going to the source of the water. So in fact, this antibiotics coming to the humans. Antibiotics being detected in the milk, because giving as a growth promoter, not as a therapeutic method, they're going down to the milk, and then it's coming to the humans. So, and they was using in the plant protection, there was, you know, if you take antibiotic, it needs to take at least 5 days to be sure that you kill the pathogens, not just reduce the number, you feel better, okay, I will stop the antibiotic. No way. All around the world these days, antibiotic for the human application is still under control, because this is the point. Need to be used only when it's needed. And several mechanisms being developed by, actually they're not developed by the microorganisms. They was always existing, but now with use of the antibiotics, this mechanism become more and more expressing, because bacteria need to be protected. They try to protect them from the antibiotic, and they will try to find a way to protect. When we do the antibiotic test, it's very important to know which antibiotic we need to test. Well, it's nothing better to everything that you have in the lab to test. Well, it's good. If you test about 50, 60 antibiotics for your strain resistant, it's nothing wrong, but then it's, your supervisor will get upset with you because you spend too much money and not really needed. And we need to pay attention on this specific list of antibiotics. I'm not sure if contrast is good enough. Can you read it or not really? Sorry, but they told me I'm not supposed to stop the light. Okay, let's try to explain. You have here the different microorganisms. Here it's a different lactobacilli. You have bifido, pediococcus, leuconostoc, lactococcus. You have bacillus. You have some gram positive, gram negative somewhere. Yeah, here is enterococcus on the bottom. And then we have some list of essential antibiotics. They need to be test compulsory. So this is the vancomycin, this is tetracycline, then gentamicin, ampicillin, chloramphenicol, cannabicin. So the list is essential antibiotics. So to discover if the strain is safe, if you check, this is recommendation, European Food Safety Authority, and it's based on the expert panel. So if you check this, maybe 9 antibiotics, you're already fitting inside the EFSA recommendation. If you can check more, okay, nothing wrong, it's even better. But if you don't check these 9, reviewer of your paper most probably will ask you, okay, why you check such a hard extent list, but you don't check essential one? So just first of all, try to check these 9. You can see the vancomycin is not recommended for some of the lactobacilli. We can see why. And then some of the others, well, here the pediococcus, well, it's not compulsory. You can do it, but you see why it's not such important. But for enterococci, it's a very important antibiotic, because resistance to vancomycin in enterococci is a really big problem. Why it's not recommended for most of the lactobacilli? Because it's intrinsic resistance. They don't have a gene for this. They can have normal processes of their structure. They will be resistant to vancomycin. Because vancomycin is not an antibiotic that's supposed to kill lactobacillus. As you know, the antibiotics can work, some work on the gram-positive, some work on the gram-negative. We have good examples of, let's say, nalidixic acid. It's working on your gram-negative, not on gram-positive. And very often people do the lactic acid bacteria, check the nalidixic acid, say, yes, I have resistance to nalidixic acid. Yes, of course you have it, because this antibiotic not work on the gram-positive. Okay, if you have done, you say, I use it as a positive control to know that it's there. But again, to be considered as a multi-drug resistant, specific microorganism needs to be resistant to at least antibiotics from different groups. You detected resistance to penicillin, to ampicillin, to oxycillin, but they're from the same group. You need to have resistance to the antibiotics from different groups. And this is important point, because if you do this, they are examples from the different groups, so you can have it. And if you do just a random antibiotic, most probably you will take, it's a risk to take antibiotic from same group. And then you detect all of them resistant, but they're from same group. So this is a very important thing. Now, the problem with the antibiotic resistance, remember 1941, penicillium being discovered, 58 vancomycin, gentamicin in 97, but it's already antibiotic resistance started in 79. So took like 30 years, really short time. And this is based on this unresponsible application of the antibiotic. They was believing that we will solve all the problems. Not at all. As I say in the first, I think it was in the first lecture, we win the battle, but we don't win the war. Still, we need to fight with the pathogens. No option. They will try to exist. Their purpose is to, pathogens want to kill us because they want to survive. We want to kill them. So, but at least now we're having new generation. We have several new generations of antibiotics. But the problem in the last year, it's almost nothing coming on the market. Really nothing. The problem is too expensive. And it's pharmaceutical companies, they're happy what they have. They're happy to do some modification on existing antibiotics, but they urgently need something new. Some new mechanism, because this way that antibiotic work, they come in, they do problem on the synthesis of the cell wall, etc. But microorganisms producing enzymes, they can attack the antibiotic. They produce the surfactants that can be maybe not allow the antibiotic to attach. So obviously some mechanisms. And this is why bacteriocins are considered as a potential, they're not antibiotics, as a potential antimicrobial that may have solved the problem of antibiotic resistance, because remember, bacteriocin need a lipid 2 to attach. And if bacteria losing lipid 2, she already have a problem. So bacteria don't want to lose the lipid 2, and in this way a receptor will be always there, so the bacteriocin can go and attach to one. What time is it now? We started at 10, so now it's... Okay, 45 minutes more and we can have break. It's okay? Are you agree? As I say, it's democracy, we can vote, but then I will take the decision. Okay, so now we're having this example, Lactobacillus plantarum, isolated from papaya. So you see why I was always mentioning that I love papaya, because it's rich of the microorganism, rich of papain, enzymes, it's a very nice fruit, and you're very happy to have it, a good quality of papaya. Yeah. Listen, in Europe, don't try papaya, it tastes terrible. I mean, they collected this small one. Do you have different names for the small and the big? No. Because in Brazil, one is called papaya, the small one. The big one is called mamão. Okay, but anyway, so I saw it already on the market here. It looks really delicious when you just watch it, because it's nice, ready to be just cut it and eat it. In Europe, they collect them green and they transport for a long time. They come full of black spots on the top. I mean, it's a tropical fruit. I mean, it's not easy to transport, so eat it when you have it. So we isolated plantarum, Lactobacillus plantarum from papaya. Would be done in South Africa. So South African papaya. And interestingly, we wanted to explore the probiotic potential of this strain. So Lactobacillus plantarum appearing as a safe strain, nothing wrong, kind of friendly strain, no virulent genes. So on the perception for Lactobacillus plantarum, supposed to be safe.